So the last talk of this session is an invited talk on image activated cell sorting by Keisuke Goda. While setting up, I was reading Keisuke Goda's paper yesterday. It's like a trivia on the paper. The manuscript, I believe, it's currently under review in Cell Journal. It has like 51 authors spanning across 26 different universities. It's kind of like a really big project. Like that's how I think the groups are going now, like bigger and doing major things rather than small groups doing isolated research like in the past. So we'll hear from Keisuke Goda now.
Oh, good. Well, thank you very much for the introduction and also the invitation. So I'm from here, University of Tokyo, but the other campus. So I will talk about a kind of new type of technology called intelligent image activated cell sorting. Before my talk, let me introduce my group. So this logo, the Goda Labs logo, sort of describes what we do in the group. Basically, it's the G, and you see the spectrum and some laser pulse. It's designed by one of my former students. Yeah, so basically, it's the G, the laser pulse, or could be the cell nucleus and the spectrum. So as you can see, like we do basically laser-based molecular imaging and spectroscopy for biomedical applications in general. So for the last several years, my group has been developing a number of high-speed imaging and high-speed spectroscopy methods. And also, we've been exploiting new applications based on new technologies. So today I will talk about the recently published paper in Cell. So here's a background, the need for better cell analysis tools. So I guess most of the audience are not familiar with biology. So let me give you a little overview of what biology is. So we deal with cells, biological cells. So cells are basically the structural and functional unit of life. And there are many, many types of cells with many, many different types of functions and also applications based on the cells. And there are lots of unmet needs in cell analysis. For example, how to identify unknown cells and their functions, how to isolate cells with distinct features, and also how do the same type of cells show different drug resistance. Like some cancer cells have a resistance to anti-cancer drugs, while others don't. And also how to detect circulating tumor cells, and or how to increase microalgal biofuel production efficiency, and so on and so forth. And there are many, many, many types of applications and a lot of needs for cell analysis. So cell analysis is sort of analogous to Where's Wally problem. And I guess when you're a kid, you used to play this kind of game, right? So what's interesting about this game? Basically, you want to find Wally at high speed with high accuracy, right? And this problem is sort of analogous to cell analysis. We want to find target cells with high accuracy at high speed, but it's quite challenging. By the way, do you know where Wally is? Okay, you're good. You guys are computational scientists. Okay, good. It's here. So scientifically, we have limitations. So how do we do identification and sorting in general? So basically, we rely on optical microscopy with pipetting, the most traditional way. But this process is very time-consuming, labor-intensive, and inefficient because you have to look at every single cell under the microscope, and then if you find a good one, you pipette that cells and move it somewhere, and then do a more detailed analysis. There's an automated system called FACTS, fluorescence activated cell sorting or cell sorter. It counts and analyzes a large number of cells in real time based on scattering and fluorescence measurements. So basically you have a cell population, you mix it with fluorescently labeled antibodies, and then you flow them through this tube. And you illuminate these cells with a laser, and then you detect light scattering or fluorescent signals. And then you sort those cells based on the signals. So this is good, but it doesn't have special information, right? Basically, it's a single point measurement. So you cannot identify and isolate cells based on special metrics. So there's a trade-off between cell picking microscope and FACTS, right? So this microscope provides high content but low throughput capability. So we can find Wally, but it will take a long time. On the other hand, FACTS is a low content but high throughput technology, right? So it can analyze a lot of cells in a short period of time, but it's very difficult to find Wally because everyone looks the same. More physically, these two technologies sort of depicted in this video merit slide. So the x-axis is the throughput, cells per second, number of cells per second. Y-axis is the basically information content, cellular information content. Here, basically the number of pixels times the number of colors, number of fluorescence colors. So fluorescence activated cell sorting is here, and cell picking microscope is roughly here. And if you plot, conventional or commercially available products, and they actually fit out these areas nicely. But this is where Wally is, right? We want to find Wally here, not here. Wally is not here. Wally is not here, right? Wally is here, right? You need higher cellular information content at high speed, right? At high speed with high accuracy. That's what you need. Yeah, so more information to make the invisible visible, and also we need to eliminate enormous time and effort. So that's the technology we want to develop. And the technology that addresses that need is called intelligent image activated cell sorting that we have recently developed and published last year. So this is a published paper in Cell. And what I want you to see is the number of courses, 51 courses, and many, many affiliations. So physics, chemistry, biology, computer science, so on and so forth. So it's a great interdisciplinary team that realized this technology. So this is a picture of the development team. The number of institutions were involved in the development. Lots of people, like more than 50 people involved. So this is sort of overview of the technology. Basically, we start with a heterogeneous cell population, and then we have a high-speed imaging capability to take a picture of every single cell. And then intelligence or deep learning analyzes each cell in real time. And then decision is sent to the sorter module, and then we identify and sort those target cells. And I have an animated movie that shows how this thing works. So this is the machine, and you put a tube of cells, and then you go into the system. And this is the big picture. The cells come in through this tube. And then cells come randomly, so we use a hydrodynamic focusing to focus every single cell into a single stream at high speed. And we have a sort of specialized high-speed fluorescence microscope to take pictures of every single cell in 3 colors. And then we use acoustic focuser to maintain focusing. And then detected fluorescence signals are digitized and sent to the image processor. And in the image processor, we reconstruct those images in different colors, and then we patch them together. And then on the image processor with deep learning, we basically extract features, molecular features, and then based on that information, we map those cells into a high-dimensional space, and then we identify which ones to sort or which ones not to sort. And then decision signal is sent to the sorter module, and then when the target cells come, we pick the cells from the mainstream, and then we eventually get sorted and unsorted containers. And this is the configuration. So we run this system based on a single microfluidic chip. So this is where cells come in. And here we measure the speed of the cells, and also we image every single cell in 3 colors. And then the information is sent to this real-time intelligent image processor, which consists of many CPUs and FPGAs. So in real time, we process the image, construct, and then analyze. And the decision signal is sent to the this dual membrane push-pull cell sorter to activate the cell sorting module when the target cell comes. So cells propagate at basically about 1 meter per second, and this distance is about 32 millimeter or 3.2 centimeter. So we have about 32 milliseconds to process everything and make decisions. And this is a picture of the instrument. It's basically an optical system combined with microfluidics and lots of computational stuff. These are the monitors to monitor each process. This is an enlarged view of the instrument. So this is a microfluidic chip. So cells come in through this tube, and they propagate within this chip. And this is a kind of pair of object lenses to image those cells. And then this is about 3.2 centimeter. And this is where we sort cells based on the additional signal. So how do we get, how do we take pictures, fluorescence pictures of every single cell? We use the technique called frequency division multiplex microscope. It's kind of complicated, but let me sort of explain how it works. The key is to generate this so-called frequency comb, especially frequency comb beam. The CW laser is split into 2 arms, and we combine via a pair of acoustic optic deflectors. So each acoustic optic deflector creates this kind of divergent beam with frequency modulation. And then when these 2 beams are mixed, we have a frequency comb, spatially, but each frequency comb line is modulated at a different frequency. But this is a frequency picture. And then this beam is illuminating the flowing cells, but each comb line is incident onto a different locations of the cell, right? And then when the cell comes in, the fluorescence signal comes about, and the 3 detectors to detect 3 different colors. And so the signal is coming in the time domain, but you know that which frequency modulated signal come from which spatial coordinates, right? Then after the measurement, we take the Fourier transform, and then we demodulate that time domain signal to get a fluorescence signal at each spatial point, and then we can patch them together to reconstruct the fluorescence image. And this is sort of the schematic of the real-time intelligent image processor, because it's a microfluidic chip. And basically, 6 signals come in, speed signals and image signals come in. And then on the IC node, we reconstruct fluorescence image. And on the IA node, image analysis node, we perform a real-time execution of a CNN, or convolutional neural network. And then everything is connected on the 10G all IP network. IP is the internet protocol to speed up the communication between the different FPGAs and CPUs. And then everything is done within 32 milliseconds. and the decision signal is sent to the solder module. And this is a microfluidic chip. It basically consists of three components: hydrodynamic focuser, acoustic focuser, and cell sorter. The cell sorter is this. Basically, it consists of two membranes, two glass membranes with PZTs. So when the target cell comes, we activate this membrane by pushing, well, by activating a PZT to push this glass membrane to create a perpendicular flow. And then this cell is kicked in this direction, or we can do this in the other direction as well. So this is sort of the demonstration of cell sorting. And I have a movie that shows the operation of this instrument. This picture, and before the experiment, we discuss and prepare for cells. And then we put a tube containing a sample of suspended cells into this machine, and then we run it, and then we monitor every event. These are cells, and these are cells monitored, and then basically we wait for some time, and then eventually we collect the sorted and unsorted cell tubes, the two tubes, and then after that those cells in the tubes are inspected under optical microscope to evaluate the sorting yield and purity. And then finally, we discuss the results. They're laughing, but they're actually discussing. Yeah. And here's a sort of basic performance. So this graph shows how much processing time it actually takes. The x-axis is basically the events ordered in the sort of the amount of time it took. The y-axis is the processing time. Under classical image analysis, pretty much all the events are within 32 milliseconds. With deep learning, we use this 8-layer CNN. It takes a little more time because deep learning takes more computational time. So then, but still 99.8% of the events are within 32 milliseconds. And based on this, we achieved the throughput of 100 events per second. Event means single cell or cluster cell or cell debris or many things could flow. So 100 events per second with 99% purity. And we tried a different set of species to make sure that this technology is broadly applicable. So we try a diverse range of microalgal cells from chlorella to euglena. And as you can see, we get 3 colors: bright field, 2 fluorescence colors, and merged image. And also we try the blood cells, and also we try cancer cells. So this technology can image a diverse range of cells with different sizes from 3 micron to 30 micron. So if you plot the specifications of this technology, actually we are here. So for the first time, we conquered this unexplored area. And if you compare that technology with a cell picking microscope, it's 1,000 times higher in throughput out of time. Okay. And we demonstrated 2 applications based on the technology. The first application is this Chlamydomonas. It's a microalgal species that lives in ponds and lakes. It's very good for biofuel research and also for studying photosynthesis. And these people, collaborators, want to study the so-called CCM, carbon concentration mechanism. It's a kind of mechanism that fixes CO2 in the body. It's very difficult to study this because it's very difficult to isolate especially unique Chlamydomonas mutants. So we use this technology to isolate those unique cells. So we basically start with Chlamydomonas cells with random mutagenesis to create a diverse pool of mutants, and we isolated about 1% of those mutants that have very strange CCM, carbon concentration mechanism. Basically, carbon is usually fixed, localized within the cell. This is a normal one. But the super ones have sort of distributed carbon fixation mechanism. This has a higher capability of photosynthesis or fixing CO2 in the body. And if you try to do this by hand or under the microscope, it will take 6 months. But we did it within 40 minutes. The next application was with blood cells. And we focused on platelets. So platelets are sometimes ignored or overlooked by a lot of scientists because they have little to do with cancer. Many people pay attention to cancer. But platelets are extremely important because in Japan and in many other advanced countries, there are lots of older people. In older people's body, you have damaged veins, blood veins, and you have a lot of platelet aggregates. They're usually good to stop the blood bleeding, but sometimes the negative consequence is that they get stuck in the heart or in the brain that lead to heart attack or stroke. So you need to isolate those, identify and isolate those platelet aggregates from the blood, but it's been very difficult. So we tried. We basically drew blood, and then we constructed 8-layer CNN to identify only platelet aggregates and isolated them, and we did the gene analysis. And if you try to do this, if you tried your graduate student or a clinician to do it, it takes 1 to 2 days, but we did it in 1 minute. So now we can do this clinically. So it received a lot of media attention. The feature, sorry, no. So we started a company. It's called Cyber. Cyber stands for cell in Japanese. It's also like cyber, right? And we're trying to develop this kind of prototype within a year or two. Please purchase it. And eventually, we want to do sort of the Rosetta Stone single cell analysis. Rosetta Stone is basically a Greek stone or Egyptian stone. It's written in 3 languages, but it basically talks about the same thing, but in 3 different languages. This is sort of analogous to cell biology. We use 3 technology conventionally: microscopy, flow cytometry, and sequencing. And their definition of biological cells are different because basically the definition of cells are based on different technologies. We need to unify these technologies to have a single language, a single definition of biological cells. So with this technology, image-activated cell sorting, we unify these 2, and eventually we want to unify all those 3 different technologies to eventually have a single unified definition of biological cells. Finally, I'd like to acknowledge the funding agencies and my group members, and thank you very much for listening.
Amazing work, Professor Gota. Unfortunately, we don't have time for questions, but then next we have break, so I'm sure we can meet Professor Gota and ask our questions directly to Professor Gota. Maybe we can take like one question if somebody has like a dying question. Srinivas.
Quick questions about clarification mostly. If you want to increase the throughput, instead of having just a single file cell going by, are there ways to look at clusters of cells heterogeneously and then have a more complex sorting mechanism? Because images can be captured parallelly, right? It doesn't have to be a single cell.
Yeah, single cell and clusters also, yes.
So it's not right now I see that it's a single file, right? One cell behind each other.
No, actually, we actually try platelet aggregates. Platelet aggregates are basically aggregates of cells.
Ah, I see.
So for example, like these are cells, but aggregated. And we image those aggregated cells or clustered cells, and then we only isolate those clusters.
How long does it take to fill up a tube?
From the start, it takes about 1 minute from the start. You insert the tube, and then it sucks up, the machine sucks up the liquid, and it's about a few minutes from the tube to the microfluidic chip. Everything can be done within like minutes, less than 30 minutes.
Thank you, Professor. Let's thank him again.
Thank you.
Let's take a break.